Hello, my name is Tiona Troung, and in June of 2024, I worked with Dr. David Chelation and Miss Kathy Schultz at the Children's Hospital Oakland Research Institute for the UCSF Summer Student Research Program to conduct hematology research. Today, I'll be presenting my project titled Comparing the Efficacy of Combined Iron Chelation Treatments with In Vitro and In Vivo Models. To give a little bit of background information on my project, sickle cell disease and thalassemia are major hemoglobinopathies, which are genetic blood disorders resulting from abnormal amounts or mutated structures of hemoglobin, which is the protein in our red blood cells responsible for carrying oxygen throughout the body. Individuals with these disorders often rely on regular blood transfusions as part of their treatment to replace their blood cells with healthy ones. However, regular transfusions may introduce excess iron into the body, leading to iron overload as a complication. If not properly managed, the excess iron could accumulate in vital organs such as the liver and heart, causing long-term damage to the body. In order to mitigate iron overload, iron chelators have been used, which are drugs that bind to excess iron and help remove it from the body. Desvril has been considered one of the most effective chelators, though it requires intravenous administration, which can be burdensome for patients. More recently, two oral chelators, L1 and XJade, have been introduced, offering greater convenience. While these three single-agent therapies are commonly used, there is a growing interest in combination chelation therapy, which involves using two or more chelators together to enhance iron clearance, particularly in cases of severe iron overload. In addition to improving efficacy, combined treatments may also increase patient compliance, making long-term management more accessible and sustainable. Because of this, additional research is needed to model the effects of combined chelation therapies to better understand their safety, efficiency, and clinical applicability. To model this, it's first important to use in vitro studies, meaning experiments conducted outside of a living organism in a controlled laboratory experiment. In this context, it allows us to examine how different iron chelators interact with iron, either free in the solution or bound within proteins such as ferritin. To evaluate the effectiveness of iron chelators, I used a Ferrine assay. Ferrine is a colorimetric reagent that binds to iron, and it forms a blue-colored complex with it. The intensity of this color can be measured using a spectrophotometer, allowing us to determine the amount of free iron in the solution. By comparing the absorbance levels with different chelator combinations, we can assess how much iron was removed from the system to determine which combinations, if any, are more effective than a single chelator alone in reducing iron levels. Figure 3 shows how Ferrine absorbance increased with increasing concentrations of iron, meaning it can be used to determine iron content. When iron chelators are added, they compete with Ferrine for binding to iron, leading to a decrease in absorbance. This effect is shown in Figure 4. It can also be seen that there is a clear order of strength of the chelators, with Desvril being the most effective, followed by L1, and finally XJade. When combining Desvril and XJade, and when combining Desvril and L1, the curve fell between those of the individual chelators. The same can be seen with the combination of L1 and XJade, as well as when all chelators were combined. Thus, in this model, no additive synergistic activity was observed. However, iron in the body is stored mostly within a protein complex called ferritin, so it's also essential to evaluate how well chelators can remove iron from ferritin-bound sources. To do this, I added different combinations and concentrations of chelators to ferritin. Using spin filters, I separated the chelator iron complex from the intact ferritin in order to measure the amount of iron that had been successfully removed from ferritin by each chelator combination. Iron content was quantified using an inductively coupled plasma optical emission spectroscopy, which is highly specific and can accurately identify elements at low levels. Desvril and L1 had similar chelating activity for ferritin in this experiment, whereas XJade had minimal effect. This data is interesting because it displays a different strength order for the individual chelators in comparison to the free iron experiment from before. This difference highlights how the form of iron, free versus protein bound, can influence chelator performance. I am currently in the process of continuing this research this summer and testing the remaining chelator combinations. Given this trend, I hope to better understand the mechanistic differences in how each chelator interacts with ferritin-bound iron and whether certain combinations may show synergistic effects in removing iron more efficiently. Current clinical trials, or in vivo studies, are being conducted to evaluate the safety and effectiveness of combined iron chelation therapies. These trials are happening alongside laboratory experiments such as this because clinical outcomes and molecular mechanisms must be studied in parallel. While clinical trials can provide crucial data on patient responses and side effects, lab-based studies help explain the underlying biological mechanisms. Taking a look at the difference mean between current clinical studies, combined chelation therapy appears to be more effective in reducing iron overload in patients, as shown with each combination displaying varying degrees of success. This demonstrates how combining chelators can lead to more comprehensive iron removal than monotherapy alone. My project contributes to this larger effort by helping uncover how iron is mobilized under different conditions, which can inform future clinical decision-making. The next steps of this project is to complete the testing of all chelator combinations with ferritin and to test chelators in human liver cells in order to evaluate their effectiveness in a more biologically relevant system. This would help bridge the gap between in vitro biochemical assays and real-world clinical outcomes, ultimately contributing to the development of more effective and personalized treatments for patients with iron overload. Thank you all for listening to my presentation.